In today's video, we'll be talking about what was the first encoded information in DNA. Well, in RNA. The first the first encoded genetic information. Hi, my name's Eduardo Hernandez. I'm the creator of Emergent Chemical Evolution, the newest theory in the origin of life. I'm also the author of the book by the same title. You can find it on all major online retailers. Well, let's get to it. Being that the ocean, as I said in previous videos, would have been similar in composition and an environment as that of a cytoplasm of a cell, that creates an inducive environment for organic materials to react and to cause replication of the first RNA molecules. Now, as I stated in the previous video, viroids, which still exist today, are vectors, viral vectors that do not have any encapsulation. They're just a long strip of RNA, and they easily infect other cells, invading all the way up to the nucleus where they are reproduced. The thing about viroids, of course, is they don't have any genetic information. They are random strips of RNA that just achieve a certain amount of length that allow them to take a conformation, wrap themselves up, and allow them to survive in the environment, which is a great ability to have at the beginning of life, which would make them more robust and able to survive until now. One of the first things that happens that we learn about in the replication of genetic information is that no system is perfect. And trust me, at that time, no genetic system would have had any type of correcting mechanism, any type of error detecting system that would have corrected any errors in the replication system, which most advanced organisms do have, but are not evident in the simplest of cells or the simplest of viruses. The level of complexity and error checking does occur in certain viruses, albeit very simple systems, I think powered by evolution. But let's get back to the purpose of this video. Errors in replication would have been the driving. Well, errors of replication are the driving force of evolution. Period. I mean, even today in us advanced animals. So it definitely would have been present in the first molecules that were replicating. And RNA is notorious for introducing errors in its replication. That's why many of the viruses that exist today that infect us are so evolved so quickly and are so hard for our immune system to constantly identify all the time, because they evolve so quickly, they change so quickly. They're basically a different virus when our immune system sees them. So that definitely would have occurred at the origin of life. And that being said, the viroid would have made replication its main purpose, and those viroids that would have replicated easier would have been the ones that would have propagated more, and those would have been propagating more errors. The errors that are common are deletion, removal, multiplication, adding, and and um, and of course changing of sequence, but which could occur with multiplication or deletion. But what we need to focus on right now, what would have made RNA more what would have been an evolutionary advantage for these strips of RNA would have been RNA that was long enough to produce well, part of my theory is that almost all biomolecules would have been randomly produced. Some form of biological proteins, polypeptides, would have been available. The ones that replicate RNA have homologs today would have definitely had homologs then. There would have been homologs, similar proteins that do the similar work of all biological functions. We've had the entire surface of the world producing trillions upon trillions of polypeptides. There had to have been ones that mimic the the biological functions of at least of the simplest life forms that exist today, or then I should say. So what would have been necessary? Just proteins that would have replicated RNA, proteins that would have produced precursors of RNAs, proteins that would have then taken those precursors and made full RNA molecules. So that would have been the norm at the time. At the beginning, you would have had RNA producing proteins that would have produced trillions upon trillions of them. Then you get the cyclic chaos engine taking those RNA molecules and propagating them. So what's the next step in an evolutionary environment would have been for which molecules would have more easily reproduced. Maybe certain sequences of RNA would have been easier to unravel and therefore easier to replicate, where other conformations would have been a more rigid twisting and conformation, where those would have not been as easily reproduced and therefore would have not as propagated in the environment. So certain things like that would have started evolution. Evolution is not a thing that you can say it's a force. Evolution is a set of rules that apply to systems that replicate. So once you have replication, or once you have the production of units that can propagate an environment and that are subject to that environment, you can create a simple form of evolution. And that would have taken place at that time, where you would have certain RNA molecules being more present in the environment, therefore having more opportunities to further evolve and further gather nucleotide length and eventually nucleotide information. Now, what's important to realize is that one of the key bits of information that are required in any organism is the replication and translation of nuclear information. One of the basic things that any nuclear system, any nucleic system needs is the translation of the information in the genome of an organism into protein. And the first things that do that are transfer RNA. Is the key component of any system like that, is the ability of that information to be translated into a mechanism that can gather the amino acids necessary to string them together. And of course, I identified in previous videos that viroids, the structure of viroid sequences mimic those of ribosomes. If you multiply the genome of viroids enough, you can have the conformation of a ribosome. I believe, well, I know for sure that viroid information is found in the junk, quote unquote, junk DNA of telomeres in advanced organisms. Sequences have been identified that match viroids. Those viroid sequences are also found in other genetic information. So there is evidence that viroids were present in our genetic past. So one of the things that has been identified in aging cells is the loss of telomeres, which are the end caps of chromosomes. So to me, that points that telomeres, which are sequences of sequence that contain viroid information, could point to the origin of life, at least in my eyes. But that's not where, that was something I eventually discovered. What I did recognize when I saw the sequence of viroids for the first time is that, again, I recognized the pattern of the conformations of RNA to match those of ribosomes, and to match the structure of transfer RNA. Transfer RNA is a strip of RNA that gathers the amino acid and brings it to, that's transfer RNA. Transfer RNA takes a specific conformation that matches ribosomes and brings an amino acid, which that ribosome takes, removes the transfer RNA, and keeps the amino acid and attaches it to another amino acids. Now, that marriage of the transfer RNA and ribosome, how did that occur in the first place? What force, what mechanism would have allowed for that to have occurred randomly? That is possible. I'm not saying it's not. Almost anything is possible. But what would be easier? If you had a strip of RNA that you can, at one point, multiply to make a larger molecule, and at the same time that strip of RNA be reduced and become a transfer RNA. Because the transfer RNAs that are created in the different kingdoms of life have different pieces of extra RNA that need to be cleaved off by the organism in order for that RNA to work So when it's replicated, the information for the RNA for the transfer RNA is longer and takes a much more convoluted configuration. And in order for that transfer RNA to work, that portion that is excess needs to be cut off. Now, that excess portion in different kingdoms of life are located at different locations on the transfer RNA, are removed in different ways. So that to me shows that there was random means of achieving transfer RNA. And if you start from viroids, which are informationless pieces of RNA that can replicate at astonishing rates, and that have the conformation of ribosomes and transfer RNA, which are smaller RNA segments. They are between 75 and 95 nucleotides. The viroid is 395, give or take, depending on the viroid. And ribosomes are much larger RNA sequences. I don't know. It depends on whether they're cellular, viral, animal, or plant, how large those ribosomes are. But they are made of different pieces of protein. So depending on where in the evolutionary scale you look at ribosomes, depends on how large they are. But you have the simplest ribosomes that are much smaller, and there have been experiments where the ribosomes are stripped of their proteins, and only left with the RNA segment, and translation still occurs. Those proteins are there for error checking and certain other functions, but just having the RNA alone still works to cleave and remove the transfer RNA from the amino acids. So that to me straight points to viroids being the initiating RNA strip, and the information for transfer RNA and ribosomes would have been the first genetic information that would have been stored. No, scratch that. Okay, I'm gonna have to edit that out. No, I'm losing it. No, the first information that would have been encoded in viroids. No. I mean, no. It had to have started with a system of translation. A system of translation would have needed to be readily available at the beginning of life. If the oceans mimic the cytoplasm, what would have been necessary for genetic information to be translated and the encoding of proteins would have taken place. So you would need, viroids would have been first, become first present because they're random pieces of RNA. From that, they would have autocleaved themselves to form transfer RNA. Then they would have had errors in replication that would allow them to multiply, have multiple copies of themselves, and create in the environment ribosomes, simple ribosomes, which would have then could have reacted with other strips of RNA, other viroids, and you would have had random lengths, random sequences of RNA that eventually one of those sequences could have become translated by those components of other viroids, and therefore become one of the first RNA, genetic information containing RNAs, where the RNA itself would have been translated directly into protein by the ribosomes and the transfer of RNAs coming together and translating that strip of RNA. That protein would have been random types of protein. What would have then created an evolutionary advantage for any of those strips of RNA would have been to first, like I discussed earlier, to have a conformation that would have been easier to unravel and allow replication of itself. Then it would be containing information that would have been beneficial to itself. Maybe the information to create a protein that replicates itself. That would be an advantage. Also, create proteins that could encapsulate its RNA and give, not encapsulate it at first, probably allow it to attach and then give it some kind of replication advantage or give it some kind of survival advantage by being attached to a protein. And then as time went on, that protein information could change, making it further protect the RNA and or give it some kind of, that RNA strip, some kind of advantage by providing a protein that aids in its replication. So you can have multiple pathways to gaining advantageous nucleic information in the RNA strips. So you'll have some that translate proteins that attach to the protein, the RNA, and give it some kind of additional robustness to allow it to survive in the environment, or translate a protein that would aid it in its replication. So that would garner some genetic information in that strip of RNA, giving it also an advantage, making it more present in the environment, giving it more opportunities to replicate and to gain further protein information. So you would have multiple ways of genetic information to be acquired in the strips of RNA. But again, my take is that the first genetic information would have been information necessary to create transfer RNA. And then other RNA sequences multiplying themselves and having multiple copies of its genome to create in the environment viroids. And then because of their origin being of the central viroid, making them complementary to each other, of having the original source, then they would more readily interact and create a system of replication. So the first genetic information had to have been the creation of transfer RNA and the multiplication of the viroid genome to create ribosomes. Now here's the thing, and this is key, this is what also led me towards viroids, because as I was researching viroid information contained within transfer RNAs, ribosomes, and telomeres, I was looking at how modern cells translate the genetic information to create transfer RNA and ribosomes. And what I found out is that transfer RNA in modern cells are produced, replicate into a long strip of RNA that then is cut into pieces by another RNA strip. It's an RNA enzyme. It's a 300 plus strip of RNA that then comes and cleaves the longer strip of RNA to create 3 pieces of transfer RNA. That perfectly fits the origin of life theory that I have that would state that viroids would cleave themselves and apparently cleave other pieces of RNA to then create a system of replication that allowed RNA to replicate and propagate in the environment, in the cytoplasm of the ocean back then. The ocean would have been a very stable system for billions of years because of the carbon dioxide laden atmosphere creating the pH gradients. And pH gradients do assist modern cells in becoming more robust and being able to replicate much better their proteins. So the first genetic information had to be transfer RNA in some ribosomes. In some viroids would have been transfer RNA information, and others, they would have gone into multiplying themselves into larger pieces and creating ribosomes. So that is the first genetic information that would have been produced at the origin of life. Then, what would benefit those RNA strips once a system like that is created? What would be the next step in the origin of life? That'll be the next video. Thank you.